Deep within the human form lies an astonishing secret, a symbiotic empire of microscopic beings. They dwell in every crevice, shaping health, dictating vitality, and even influencing our innermost thoughts. This unseen population outnumbers human cells by a staggering margin, forming complex, thriving communities that have co-evolved alongside our species over millions of years. For millennia, their presence remained entirely hidden from human consciousness. Modern science now reveals that these countless organisms are not dangerous invaders, but essential biological partners. They are fundamental to what it truly means to be alive. From the very moment of birth, a complex biological exchange begins, laying the foundation for our internal living landscape. Each human being is not merely an individual, but a living, dynamic ecosystem. This intricate web of life, collectively known as the human microbiome, includes thousands of species of bacteria, viruses, fungi, and ancient archaea. They thrive in specialized niches throughout the body, maintaining a delicate equilibrium that defines our unique biological identity. The largest and most diverse of these microbial communities resides deep within the human gut, a vibrant metropolis of cellular activity. Here, trillions of organisms work ceaselessly day and night. These microscopic residents perform vital chemical functions that human cells simply cannot accomplish alone, acting as an indispensable organ of survival. They break down complex plant carbohydrates, synthesize essential vitamins, regulate metabolic rate, and fine-tune our immune responses. Without them, the human body would struggle to extract vital nutrients from food. This documentary explores this hidden world within, revealing how these microbial companions shape our physical health, emotional mood, and genetic expression. Prepare to redefine what it means to say I. The sheer scale of this microbial universe is difficult to comprehend, yet it profoundly impacts every single aspect of human biology. Its far-reaching influence extends beyond mere digestion, touching the structural core of our neurological, cardiovascular, and immunological existence. The human body is not a solitary entity, but a walking, breathing superorganism, a holobiont forged through biological cooperation. Every square centimeter of our skin hosts millions of specialized microorganisms, forming a living armor against foreign pathogens and harsh environmental stressors. Even the mouth, serving as the gateway to our digestive system, harbors hundreds of distinct bacterial strains that contribute to oral ecology and the initial chemical breakdown of food. Meanwhile, the nasal passages and delicate respiratory tracts maintain specialized microbial communities that act as frontline defenders for every breath we take. However, the gut microbiome remains the undisputed star player, a vast internal rainforest teeming with genetic diversity. Billions of bacteria, fungi, and viruses coexist in intricate balance within the intestinal tract, creating a dense biological community that rivals the human brain in both weight and metabolic complexity. Each individual's microbiome is as unique as a fingerprint, sculpted by genetics, early life events, dietary habits, and environment. No two microbial profiles on Earth are identical. While beneficial microbes work tirelessly to protect us, opportunistic species lie in wait, ready to expand if the internal balance is disturbed. The constant equilibrium between these microbial factions dictates much of our resilience against disease. Maintaining this delicate harmony is paramount. This internal ecosystem is endlessly adaptable, dynamically responding to every meal consumed, every prescription taken, and every surge of physical or psychological stress encountered in daily life. For instance, dietary fibers and resistant starches feed beneficial bacteria, allowing them to proliferate and produce protective compounds. Conversely, a diet dominant in ultra-processed foods and refined sugars starves these friendly microbes, fueling inflammatory species and triggering cascading metabolic disruptions throughout the entire body. Decoding this internal landscape is revolutionary for modern medicine. We are only beginning to grasp its vast genetic potential. The collective gene pool of our microbiome contains over 3 million unique genes, outnumbering human DNA by more than 100 to 1, providing an astonishing reservoir of biological intelligence. This vast second genome grants us incredible metabolic versatility. Each microbial strain brings specialized enzymatic tools, expanding our ability to synthesize compounds, neutralize toxins, and processed nutrients. They are tiny biochemical powerhouses working around the clock to sustain our complex physiological needs. A primary responsibility of gut microbes is extracting hidden energy from our diet. They ferment complex carbohydrates that human digestive enzymes are completely incapable of breaking down. Without these microbial partners, valuable fiber would pass through unutilized, depriving our cells of crucial metabolic fuel. Through fermentation, microbes transform fiber into short-chain fatty acids like acetate, propionate, and butyrate. Butyrate serves as the primary energy source for colon cells, strengthening the intestinal wall, mitigating systemic inflammation, and even regulating cellular growth to guard against disease. Furthermore, these organisms act as an internal pharmaceutical factory, producing critical B vitamins, vitamin K2 for bone and vascular health, and essential amino acids. Simultaneously, the immune system, 70% of which resides in the gut, relies on constant microbial signals to calibrate its defenses. From infancy, microbial molecules interact with special receptors on mucosal immune cells. This continuous conversation trains our immune system in the art of immunological tolerance. It teaches defensive cells to tolerate benign food proteins and friendly microbes while remaining hypervigilant against genuine threats. Without this crucial microbial education, the immune system becomes prone to severe miscalculations. A depleted microbiome can lead to hyperreactive responses, giving rise to autoimmune disorders, chronic allergies, and persistent low-grade systemic inflammation that quietly damages healthy tissues over time. Microbes also defend us physically by colonizing intestinal binding sites and consuming available nutrients, effectively starving out invading pathogens. In addition, many beneficial species secrete natural antimicrobial peptides, bacteriocins, that actively destroy harmful bacteria in a continuous, invisible chemical war. When this protective barrier collapses, the consequences ripple across every organ system. Beyond defense, gut microbes regulate fat storage, insulin sensitivity, and metabolic rate. Specific bacterial ratios dictate how efficiently we harvest calories, directly influencing body weight and metabolic efficiency. Fascinating research reveals that microbes can even manipulate host appetite. By producing metabolic byproducts that interact with hunger hormones like ghrelin and leptin, certain bacterial populations send chemical signals directly to the brain, driving cravings for the specific nutrients they need to survive. Astonishingly, our gut microbes operate on their own biological clocks, synchronized with our circadian rhythms. When we eat, sleep, or travel across time zones, our internal bacteria shift their metabolic activities accordingly. Disrupting this daily rhythm through irregular sleep or night shifts alters microbial output, impacting glucose tolerance and energy balance. This intricate interplay reflects eons of coevolution. We are not solitary organisms operating in isolation, but integrated biological networks. Our survival has always depended on this cooperative alliance, where human host and microscopic resident share a unified biological destiny. This radical perspective challenges classical biology's definition of the self. The boundaries between human host and microbial symbiont are fundamentally fluid. Far from being passive hitchhikers, these microbes actively modulate our physiological pathways through an ancient, refined cellular language. Unlocking this cellular communication is transforming modern therapeutic strategies. Nowhere is this connection more surprising than along the gut-brain axis, a complex bidirectional communication highway linking the gastrointestinal tract directly to the central nervous system. The primary physical conduit of this axis is the vagus nerve, a massive neural cable transmitting signals between gut and brain in real time. Gut microbes produce over 90% of the body's serotonin and a significant portion of dopamine, key neurotransmitters that regulate mood, sleep, stress response, and emotional stability. Gut microbes release these neuroactive molecules. They stimulate local nerve endings, sending immediate signals to the emotional centers of the brain. Clinical studies confirm that imbalances in gut flora correlate strongly with clinical depression, generalized anxiety, and neurodevelopmental conditions. The foundational blueprint of this neuromicrobial ecosystem is established early in life. During vaginal delivery, a newborn is coated in maternal bacteria, receiving its first vital microbial inheritance. In contrast, infants delivered via cesarean section miss this initial exposure, acquiring distinct bacterial strains from the hospital environment. Maternal breast milk further nurtures this developing microbiome. It contains specialized complex sugars called human milk oligosaccharides, which infants cannot digest. These sugars exist solely to nourish specific beneficial bacteria, like bifidobacteria, ensuring the baby's intestinal lining is rapidly fortified. As children explore their world, contact with animals, soil, and diverse whole foods continuously expands their internal flora. Early childhood exposure to environmental microbes builds ecological diversity, building a resilient immune system capable of resisting modern inflammatory diseases. Regrettably, modern lifestyles have severely disrupted this evolutionary contract. Over-sanitization, urban isolation, processed diets, and sedentary habits have created an internal biodiversity crisis, stripping away microbial species that protected our ancestors for generations. Antibiotics represent one of medicine's greatest achievements, saving millions of lives. Yet broad-spectrum antibiotics act like a forest fire within the gut, indiscriminately wiping out benign and beneficial communities alongside pathogens, often leaving long-lasting ecological scars. This profound state of microbial imbalance, known as dysbiosis, is now recognized as a major underlying driver of modern chronic illness. When beneficial species disappear, opportunistic organisms colonize the void, releasing toxic metabolites that destabilize host physiology. Diets heavy in refined carbohydrates, artificial additives, and chemical preservatives further starve helpful microbes while eroding the gut's protective mucus layer. Combined with chronic psychological stress and sleep deprivation, our modern environment creates a perfect storm for internal deterioration. This breakdown degrades tight junctions between intestinal cells, causing leaky gut syndrome. Microbial fragments and uningested food proteins seep into the bloodstream, triggering systemic inflammatory cascades implicated in inflammatory bowel disease, metabolic disorders, and chronic fatigue. Metabolic syndrome, type 2 diabetes, non-alcoholic fatty liver disease, and arterial plaque formation are all closely linked to dysbiosis-induced inflammation. The health of our microscopic companions directly dictates the structural integrity of our cardiovascular and metabolic systems. Even neurological health suffers when the intestinal barrier is compromised. Inflammatory cytokines generated in an unhealthy gut can cross the blood-brain barrier, contributing to neuroinflammation, cognitive decline, and accelerating neurodegenerative conditions like Alzheimer's and Parkinson's disease. Recognizing these destructive pathways has ignited a medical revolution. Researchers are uncovering key keystone species, such as Akkermansia mucinifila, which fortifies the gut lining, and Faecalibacterium prausnitzii, a powerful producer of anti-inflammatory butyrate, paving the way for next-generation targeted therapeutics. Restoring our internal ecosystem requires an integrative approach. Traditional probiotics introduce live beneficial strains, while prebiotics, specialized plant fibers found in garlic, onions, oats, and legumes, provide essential fuel to help these introduced microbes establish permanent colonies. Emerging science highlights postbiotic metabolites, the beneficial compounds produced during bacterial fermentation, as direct therapeutic agents. Incorporating fermented foods like kimchi, sauerkraut, kefir, and traditional yogurt introduces both active microbes and their valuable metabolic byproducts into our digestive tract. Lifestyle interventions are equally potent. Regular physical exercise promotes microbial diversity, while stress reduction techniques lower cortisol levels that otherwise damage mucosal lining. Adequate deep sleep allows the gut barrier to repair and recalibrate its daily metabolic functions. In severe medical cases where dysbiosis causes life-threatening infections, like Clostridioides difficile, fecal microbiota transplantation has demonstrated astonishing cure rates exceeding 90%, proving that replacing an entire degraded ecosystem can instantly restore host health. We are entering the era of personalized precision medicine. High-throughput DNA sequencing allows clinicians to map an individual's gut microbiome in exquisite detail, enabling customized nutritional, probiotic, and pharmaceutical strategies tailored specifically to a patient's unique genetic and microbial architecture. This ongoing paradigm shift fundamentally redefines our position in the natural world. We are not isolated organisms struggling against an external environment, but complex walking ecosystems continuously exchanging biological information with the living universe around us. To care for our health is to care for this vast internal wilderness. By nourishing our microbial partners through conscious choices, rich plant diversity, clean environments, and balanced living, we cultivate an enduring sanctuary for vitality, longevity, and mental clarity. The revelation that half the cells within us belong to non-human life forms is not a threat to our identity, but a profound celebration of evolutionary unity. We are living proof of life's infinite capacity for collaboration, thriving only through mutual interdependence. As science continues to peel back the layers of this microscopic frontier, one truth becomes undeniable. We do not walk through nature alone. We carry an entire cosmos of nature within us. Embracing this unseen world is the key to unlocking the future of human health, opening a doorway to true biological harmony.